My presentation is on osteoarthritis. Some background information about osteoarthritis. This is the most common form of arthritis, which is a type of degenerative joint disease. It is also often called wear and tear arthritis. Symptoms will occur when the protective cartilage, otherwise known as the articular cartilage, wears down over time. This cartilage helps cushion the ends of bones during movement and absorbs impact on the bones. The photo here shows a healthy knee joint and one with osteoarthritis. The meniscus is a C-shaped cartilage padding for the knees sitting between the tibia and femur. Each knee contains two menisci, which is also seen here, and bone spurs, are also known as osteophytes, are the extra bone tissue growth that are usually unproblematic unless they irritate soft tissues, interfere with joint movement, or cause pressure on nerves. For the epidemiology, these are the main factors which contribute to the prevalence of osteoarthritis in populations. OA increases with age due to the general wear and tear of the joints, and OA also increases with obesity due to overload and excessive pressure on the joints. Gender and hormones play a role as well, with women more likely to develop OA with greater severity, and this occurs generally after menopause. Race and ethnicity, genetics, diet, previous injury and surgery, and occupation are also all factors that can lead to the development of OA in a specific population. OA can be detected pathologically, radiologically, which is the standard, or clinically, and this disease is graded on a scale of 0 to 4. The prevalence of OA between genders is still in research. However, some explanations for this are the basic anatomy. Wider hips are seen in women, and this can cause a greater load compared to men. But limb alignment itself is not a sole factor for the observed difference of OA prevalence between women and men, because OA also increases around the time of menopause when women enter their 40s and 50s. It is also thought that hormones play a role. Specifically, the hormone estrogen, this decreases at menopause, and estrogen, which increases the looseness of knee joints during pregnancy and during the menstrual cycle, also has roles in inflammatory and chondrocyte metabolism, but this area is still in continued research. OA also varies between racial and ethnic groups. It was observed to be more common in the African American population than Caucasians within the US, and knee osteoarthritis was seen more in Chinese women than Caucasian women when comparing two studies. Further research is required to determine if and how anatomical features or other differences between races and ethnicities lead to the development of OA, and there are limitations in these studies due to varying protocols and standards when collecting data from different populations. OA uses the Kellgren and Lorentz system to classify its severity. Grade 0 is the absence of OA in the X-ray picture. Grade 1 depicts doubtful joint space narrowing but possible osteophytic lipping, and this is also known as bone overgrowth. Grade 2 is minimal with definite osteophytes and possible joint narrowing. Grade 3 is moderate with defined joint space narrowing in the X-ray, some sclerosis, and possible deformity of the bone ends. Grade 4 exhibits large osteophytes, marked narrowing of joint space, severe sclerosis, and defined bone end deformity. And here is another figure that shows the development of osteoarthritis from grade 0 to grade 4. For the etiology of osteoarthritis, so osteoarthritis is a multifactorial disease, so there are many causes that leads to its development, which were previously mentioned before. OA can be categorized into primary and secondary osteoarthritis, and primary OA is the most common and it has no known cause. It just develops over time with aging. Secondary OA is caused by another disease, infection, injury, or deformity, and an example of this is obesity, which would be categorized as secondary OA. Genetics plays a role in OA as well, with a larger influence in the development of hands, hip, and knee OA. Joint hypermotility, laxity, or double jointedness. This is due to weak ligaments and can lead to the development of osteoarthritis as well. There are many molecular factors that play a role in osteoarthritis pathogenesis. Inflammation is the main mechanism during OA progression. There are two central pro-inflammatory cytokines for this disease. Interleukin-1 beta is the most studied cytokine in regards to osteoarthritis. It binds to its receptors on the surface of chondrocytes and synovial cells, activating the nuclear factor kappa B, a transcription factor, and the mitogen-activated protein kinase pathways. After activation, this promotes the transcription of genes involved in inflammation and catabolism, such as the matrix metallopeptidase 1 gene, which degrades type 1, 2, and 3 collagens. The MMP13 gene is especially destructive in OA because it specifically targets type 2 collagen, which is the main type that forms articular cartilage. Communication between the MAPK and NFKB pathway enhances the inflammation response in OA. In the MAPK pathway specifically, it is activated by the inflammatory cytokines or mechanical stress often experienced with OA. This induces the upregulation of inflammatory genes, extracellular matrix degradation, and chondrocyte apoptosis through phosphorylation and activation of downstream transcription factors. The IL-1 beta cytokine also promotes the production of nitric oxide and prostaglandins, which contributes to pain and inflammation as well. The tumor necrosis factor alpha is the second cytokine central to osteoarthritis. It causes similar effects as the IL-1 beta cytokine. However, TNF alpha is specifically known to increase nociceptive neuron sensitivity. TNF alpha activates the enzyme protein kinase C alpha, which upregulates cyclooxygenase 2 expression and elevates prostaglandin E2 production. Oxidative stress from reactive oxygen species are also highly elevated in osteoarthritis. This further contributes to inflammation by activating the nuclear factor kappa B pathway and stimulating cytokines. ROS can also function as secondary messengers and cause cartilage degradation by inducing the expression of matrix-degrading proteases, reducing ECM synthesis, and also induce apoptosis. Studies with stem cells are being actively researched to discover a new treatment for osteoarthritis. Synovial mesenchymal stem cells are known to contain chondroprotective effects. This study by Long et al. Began with an interest in the regulatory potentials of synovial mesenchymal stem cell-derived exosomes. This is because past experiments have shown that exosomes, such as those derived from bone marrow, carry components for intercellular signaling, promote metabolism, and inhibit chondrocyte apoptosis. These exosomes are key vesicles that can transport molecules to target OA and could be used as a potential therapy. These SMSC exosomes used in these studies are known to have the ability to carry matrillin-3 to chondrocytes. Matrillins are cartilage matrix adaptive proteins of the cartilage ECM and released by the chondrocytes. They act as bridges between collagen II and proteolytic networks. This experiment used 8-week-old male wild-type mice. 80 underwent meniscus surgery to induce osteoarthritis, and 8 received a placebo surgery. They found that an overexpression of MATN3 prevented extracellular matrix degradation and chondrocyte autophagy defects. The results of this study shows the potential of SMSC-EXO as a drug or biomolecule carrier to treat osteoarthritis and possibly other diseases as well. You can also see in Figure E the benefits of MATN3 compared to the untreated induced osteoarthritis in a negative control tissue. In the graph next to it, the synovialitis score was also lower with the MATN3 treatment. As stated before, osteoarthritis targets specific joints such as the knees, hip, interphalangeal joints of the fingers, and spinal facet joints. The clinical presentation of osteoarthritis is pain and stiffness, crepitus, which is defined as a popping, clicking, or crackling sound of the joint due to the bones grinding against each other without cushioning, and joint deformity as seen with the hand from the accumulation of osteophytes, and joint swelling. The main goal of osteoarthritis treatments is to relieve pain and stiffness. The only way to fully cure OA is through surgery. However, there are ways to alleviate symptoms by taking oral and topical medications or through injections, such as with corticosteroids for pain relief and hyaluronic acid to improve joint fluid viscosity and cushioning. Other remedies include physical therapy, acupuncture, massage, and radiofrequency ablation, where an X-ray is used with a needle to burn the nerve ending to relieve the pain. NSAIDs are the main drugs used to alleviate osteoarthritis. There are oral and topical forms. Most NSAIDs are non-selective and inhibit both isoenzymes within the cyclooxygenase family, known as COX-1 and COX-2. COX-1 is constitutively expressed and is known as the housekeeping enzyme that maintains the mucosal lining of the GI system, among other functions. COX-2 is expressed as an inflammatory response. NSAID inhibition is competitively reversible. NSAIDs compete with arachidonic acids for binding on the active site of the COX enzymes. This binding produces thromboxanes, prostaglandins, and prostacyclin, which induces the inflammation seen with osteoarthritis. Inhibition of COX-2 enzyme specifically gives the desired NSAID effect and pain relief.